Okay, if everyone could make their way back in, we'll have our FDA Town Hall, which may look kind of strange to you right now because there's no one on stage with me. All of our members from the FDA will be joining us virtually, and I will be moderating the panel, and hopefully I have some questions we've received already, but at any point I would love for you all to come up to the microphones and ask any questions that you have of the FDA or submit them virtually. And I'll go ahead and sit over here. So thank you all for joining our special FDA Town Hall. And here is your great opportunity to be able to ask questions of the FDA directly, although virtually. And I encourage you to do so. We have several panelists from FDA's Center for Devices and Radiological Health that are joining us today. And so to start, I'll ask each of the panelists to introduce themselves and their role at FDA and how they are currently engaged with the focused ultrasound community. So I will start. Now I'm going to have to look well at the names. I'll start with Shubha Marvuda because I know her the best. So I'll start with you, Shubha.
Thanks, Jessica. Hi, everyone. Shubha, and I'm an ultrasound engineer in the Office of Science and Engineering Laboratories. I am the lead of the therapeutic.
Uh-oh.
TC87 ultrasonics. Development and maintenance of test measurement standards for medical ultrasound equipment.
Great. I think you unfortunately you cut out a little bit there.
Oh, around the standards part. I'm also involved in IEC. Can you hear me okay? Yeah.
Yeah. And then let's see. I'm just gonna look at my list here on my phone. It's easier to read. I'll now turn to Jenna Osborne.
Hi, everybody. My name is Jenna Osborne. I'm a scientific project manager in the Office of Science and Engineering Labs within CDRH. So I get to work with the ultrasound program, but also our 19 other programs as well in our research. We're the research arm of CDRH. So thank you for coming.
Great. And next, Bennett Blumenkoff.
Yes, good afternoon. I'm a neurosurgeon and clinical reviewer on a number of files for CDRH and our office, OHT5, that involve the focused ultrasound for both ablative and the microbubble blood-brain barrier disruption. Generally, my role is to review the clinical trial designs and safety and effectiveness purposes of the trials and make any concerns known, both internally and with the sponsors.
Thank you. Matt Myers.
Thanks. Yeah, Matt Myers. I'm a research physicist in the Division of Applied Mechanics, along with Shubha. I've done a lot of work in non-invasive methods for power measurement. We've done ultrasound and neuromodulation in the peripheral nervous system. We've done mild traumatic brain injury. And I am a consultant on most of the submissions that involve neuromodulation and blood-brain barrier opening. Thanks.
Now, Greg Kittleson.
Hello, again, Greg Kittleson. I work as a lead reviewer. So submissions that come into our office, neurosurgical devices, may be assigned to me or one of my colleagues. The lead reviewers will assemble a review team of subject matter experts across the various items that are relevant in a given submission. And then I would have the contact with you or one of your colleagues who've submitted a file for our review. Thank you.
Great. And Devashish Shrivastava.
My name is Devashish. I'm from Office of Health and Technology 4, otherwise known as Office of Surgical and Infection Control Devices. I am involved typically with review of tissue heating, thermal energy spread, and thermal damage spread of various devices. Thank you.
Great. So again, I have several questions that I've received that I'll start with. But again, please send your questions via the Q&A function if you're on the virtual platform, or come to the microphones here in the room at any time with your questions. So I'll start with a question around the pre-submission meetings, specifically pre-IDE submission meetings. So the question is, well, kind of a threefold question, but first, when does it make sense to have a pre-submission meeting? Does anyone want to field that?
So as a lead reviewer, I'd like to go ahead and give my perspective on this. In a sense, what comes in is going to affect anything that can come out. And so the extent of description of a planned either study or device or whatever it might be, that's really the best opportunity for somebody to get very clear comments, detailed comments from FDA. And so in terms of timing, I see it as very helpful once somebody is ready to engage on some important aspect that will really guide them through the regulatory process, be it perhaps a study design or a animal study design. It could be regulatory questions. I encourage people to be specific in what they submit and to ask specific questions, and that really leads to the most fruitful interactions.
Does anyone else have any comments on pre-submission meetings? I think,
this is Dr. Blumenhoff. I think the pre-submission from the clinical standpoint begins the dialogue to address, what are the important issues with regard to involving, people or patients in any sort of investigative protocol. And that can involve, safety concerns, that can involve effectiveness endpoints, that can involve just who actually you believe is an appropriate candidate for the study or for the device application. And it's a collaborative effort, both internally among my colleagues who may be non-clinical but have issues after they review the pre-submission, and also a collaborative between our team and the sponsor or the investigator. So it's a way to begin that dialogue and build on a foundational understanding. So then when the process matures to the point that you're ready to submit the investigative protocol, or the IDE, we call it, then it's almost like everything's been accomplished, and it's easy sailing from that point. Because at the IDE stage, we have a very, very limited time constraint, and if we can't get it resolved, then it's kind of put on we call it a disapproval, and that begins a whole evaluative process, and it's so much easier to do it up front and prior to that submission, and get all the bumps out of the road, or the nicks and crannies out of the system. So I encourage it both clinically, and I'm sure all the non-clinical really like to get their issues addressed as well.
What is the timeline for pre-submission in terms of, the time it takes for FDA, or like scheduling it, the time it takes for FDA to respond, that sort of thing? So if companies are thinking about adding that in there, the amount of time they're kind of expecting they'll be adding into their timeline.
Thank you for the question. I did want to share, I'll go ahead and put something in the chat here, but we do have a guidance document that spells out those details of the Q submission process. And so once I figure out how I can get that into the chat. In terms of the timing, 70 days after FDA has received and received a submission, a pre-submission, there'll be written comments on that pre-submission sent to the sponsor investigator. And if a meeting has been requested, that's typically scheduled on the order of 5 days after those written comments have been sent out. And then just there's an administrative thing at the front end, just in terms of being able to schedule the meetings. So typically within the first couple weeks, 15 days after some pre-sub has been submitted, it's been assigned to a lead reviewer, and I, as a lead reviewer, would reach out to the sponsor and look for a mutually agreeable date to have that meeting. Get that reference that I sent to the FDA guidance. Thank you for that.
Yeah, and we can make sure that people get that reference as well. So, you know, those of us in the focused ultrasound field know that there is a move towards a lot more combination device drug trials and products. And I think a lot of people have still some questions about how that process works with the FDA. You know, we know there's an Office of Combination Products, but I guess one, if you could sort of discuss a little bit more that process. And then we do have some companies who've indicated that they've had challenges kind of getting stuck between CDRH and CDER and really not sure how to resolve that. So maybe just, you know, kind of talking about the process around these combo products from the company perspective, you know, INDs versus IDs, and then maybe how to resolve some conflicts that could occur.
Great. In that realm of combination products, again, we've got a guidance document that came out this year. Subha, perhaps if you could put that one in the chat as well. But that really provides a lot of clear current information about the details here. But at a high level, we're always looking for the principal mode of action, mechanism of action, excuse me. So in the submission, let's say it's a topic that hasn't had some prior experience, to know for sure if it would go to the device side or the drug side or the biologic side. By submitting, again, a very clear description of what the mechanism of action is of the device aspect, of the drug or biologic aspect, and then providing either literature or perhaps some test information that gives the sponsor's perspective what the principal mechanism of action is. That would guide which of the centers would be the lead center that sponsor would be interacting with, and whether that eventually would be, let's say for a device, an investigational device exemption file, an IDE, or for a drug or biologic, an IND. And then again, as a lead reviewer, when I get these, I'm interacting with colleagues at our center who are specifically tasked in this product jurisdiction area, and they work with their colleagues at the other two centers to work this all out. So my message is, again, by placing very specific information that group will have access to as to what the sponsor identifies as the primary mechanism of action and the rationale for that, the evidence for that, that brings these right along as expeditiously as possible to the proper lead center.
This is Dr. Blumenkoff. With regard to the focused ultrasound, and specifically, more apparently, the barrier disruption indications, since the lesioning would not involve another entity. First to understand, you only have to file one or the other. So you don't need to file both as far as an ID and an IND. We emphasized this a few years ago to make sure that that was clear, because we capture, either center captures through the other process the salient concerns of safety and effectiveness. So you only have to make one submission, be that ID, which is a device submission, or IND, which is a drug or biologic. But what generally occurs, and Greg is entirely appropriate to suggest you look at all these documents, but at a practical level, just so you have a starting point, in my opinion, if it's involving a drug and it's on-label use of the drug to use in concert, if you will, with the focused ultrasound, then we get it as a device because it's an on-label drug. If it's an off-label drug or a novel drug or a novel biologic, then generally that's going to default to CEDAR or CBER, and we'll be the support on that. So it's just some way of understanding it in your own minds, whether it's more likely than not to come to us or the other center.
Great. Does anyone else have anything to add there? Okay. I think another thing, and this is sort of combo or maybe not, but there are a lot of individuals, or companies, whether it's in blood-brain barrier opening or other type of applications where they're using a microbubble or other type of particle to either enhance focused ultrasound or to enable focused ultrasound, again, to like open a barrier like the blood-brain barrier. In this case, is a bubble considered, or do you have the answer? Is the bubble considered a drug, or is it considered a device, or the microbubble, or does that vary? That's sort of a question as well.
I'll go ahead and give the general comment, so that if there is something that is clearly and explicitly simply acting in a structural context as a device, that will be a device-led submission, and such a pre-submission could be directly submitted to CDRH. If there's precedence for a particular substance that has been regulated as a drug, and then maybe can be used as a device, there may be something, as Dr. Blumenkoff was describing, as to how that gets handled. It's not crystal clear. Everything's going to be always one way or the other. And so my message is, you can always contact either of the agencies, even by email, and just start a question that way if it's that sort of a high-level question. And then that'll guide toward, is it necessary to submit a pre-submission, or can some email correspondence move that question along as to where should one be submitting something.
Okay, thanks. Any other comments there? Okay. So, again, when we're thinking about sort of the future focused ultrasound and evolving more from just an ablative application to other types of mechanisms of action, there are a lot of mechanisms of action using much lower energies, things like neuromodulation or hyperthermia. Could these be classified as class 2 versus class 3? Obviously, there are maybe not predicates, but there are things that you could compare it to, like TMS or microwave. Can those be deemed predicates from the FDA, or do predicates always need to be the same energy source? Does anyone want to tackle that?
I'm going to try to answer this. It's a very wide question. So typically, when we look at any device, we look at the risk of that device. So these are the kind of questions where, like Greg and Bennett were talking about, you do want to come in with a pre-submission. Because the question that you're asking is, one, do we consider that as a significant risk device or not? And if it is considered as a significant risk device, then there is a good chance it will become PMA or Class 3 device. If there is a predicate, or if it has been in the past, determined to not a significant risk device, that's when you will go for 510K or de novo kind of thing. But again, I'll encourage everybody to come in as quickly as possible to discuss these questions. If we are looking for the, 510K versus de novo versus those kind of questions, then I'll encourage you to go over our 510K guidance. You will see that there are a lot of questions. There's a flow chart that you can go through. And, one of those questions is, and this is regarding when, there are two possibilities. One, whatever device you have, there is another device that does something similar. If there is such device, that becomes your predicate. If there is no such device, meaning, let's say the technology is different, as this question states, then the question becomes, if the new technology or the differences in technology raises different questions of safety and effectiveness. And those are the questions you want to engage FDA with. If it turns out that they are raising different questions of safety and effectiveness, then the next question will become if it is Class 2 or Class 3. And again, you will need to have some kind of discussion with FDA to determine if it is Class 2 or Class 3. If it is Class 3, you will come in with the PMA. If it is Class 2, because there is no predicate and there are different questions of safety and effectiveness, you may have to come in with a de novo.
Okay. Did anyone else have anything to add?
Yeah, this is...
Yeah, I'm sorry, go ahead.
Okay, one of the issues that came up in using electrical energy as a predicate for ultrasound energy is that there's a new risk. Deb was just talking about risks, and that is mechanical damage that you don't see with the electrical energy. So that has come up in submissions before, and I don't frankly know how it was resolved, but that was one of the answers to the questions that Dev just brought up. Namely, there is a risk of mechanical damage associated with ultrasound that you don't see in these other types of energy.
Okay, what I was just going to say, Matt, maybe you could, or Dev can comment. I think part of that question was the same energy type, but at different maybe intensities or something. Would we view that differently? And I think there are some examples of devices that come in with a similar, say, energy component, say a laser, I'm familiar with, and there was another one I'm trying to recall, where the actual amount of energy being employed or deployed, if you will, is so relatively low that would change the device characteristic as far as risk profile. So I don't know if that would be possible or pertain, say, to focused ultrasound, but I think it is, as an example, something with some of these laser devices where the wattage is so low as compared to other lasers that they represent a different risk profile. I don't know if Dev or Matt have any experience with that. Guess not.
Okay. And I think that sort of leads to another question, which is sort of around these applications, mechanisms of action where it is very low pressure or very low intensity, and versus, say, the ablation. Would there, and again, I know that this would require pre-submission meetings and stuff, but would there be potentially times where these mechanisms of action and applications with them would not require a full FDA review because the pressure or intensity level was so low? Or is everything and kind of deemed more like diagnostic levels, or is everything therapeutic really going to need to go with the full FDA review?
Well, what that really relates to is what are we actually reviewing? We talk about the investigational device exemption, and there is what's called a study risk determination submission, which I think is something along the lines of what you're, kind of intimating that one could submit. So you have a device, and you want to use that device, and you want to understand, does it really fall under the IDE regulations for investigative protocol? And what one has to appreciate, and this is in a general sense, when we talk about a study risk determination, we're not talking about the device. We're talking about what the study is doing insofar as with the patient care or with the patient. So the example that I was given to understand that was, say, a blood pressure sphygmomanometer or a blood pressure monitoring device, where the study was to determine whether the individual was having a seizure. And with the seizure, they would get hypertensive. So the blood pressure monitoring device would alert in some respect that there's a seizure. Well, the device itself is rather benign. It's almost innocuous. But in that context of use, it could present risk to the patient, particularly if there's a false negative or something like that. So the best way to approach those sorts of questions, in my mind, is at a very preliminary stage is to submit a study risk determination submission. And actually, the IRBs of most institutions also have familiarity with that. Because if we determine that it is a significant risk, then it has to go through our regulatory process. If we say it isn't, then it does not. So it's a pretty big dichotomy between what we do have to look at and what we don't. And there are some other fine other subsets of study designs. If it's already an approved device and it's being used in a similar fashion or the same fashion as approval, often that kicks it out of having to go through an investigational protocol. So I encourage a study risk determination as the best approach.
Thank you. Did anyone have anything else to add there?
Yeah, this is Matt Myers. Jessica, I think neuromodulation is an interesting example. I think the way that most neuromodulation studies are presently conducted, FDA can move those through pretty fast and arrive at a non-significant risk decision. And so in answer to the question, yeah, FDA needs very little involvement sometimes, or a lot of the time. And that's because these are conducted at diagnostic levels. Now, there are examples or counterexamples. For example, if the patient population could be suicidal, then you wouldn't automatically call these, you wouldn't automatically make an NSR decision on that. So that's kind of where neuromodulation is now. But where neuromodulation is going, we don't really know, because right now we check for thermal and mechanical damage. But as a lot of people in the audience know, that with neuromodulation you can do more subtle things, like you can induce an inflammatory response, you can alter the way that the nerves function. Okay, so that's a kind of that's labeled damage in the literature, but there's no thermal or mechanical, there's no mechanical tearing and there's no thermal burning. It's just you're altering the functionality of the nerves, and sometimes for long periods of time. So and as you up the intensity, we would see that more and more. So where neuromodulation is headed is a really interesting and open question. But I would say right now it sort of fits the criterion of minimal FDA involvement if you're just looking for a significant risk or non-significant risk decision.
Great, that's really helpful. Yeah, I don't know if any of you were watching the sessions earlier, but you got a couple of nice shout outs from individuals that are involved in pediatric clinical trials for children with brain tumors. And I wondered if you had any just general comments on pediatric applications and maybe special considerations that you have to take in that patient population, or any additional pieces of information that that study sponsors should consider providing for you when it comes to pediatric populations where there may not be as much to go on in terms of the prior clinical experience with the devices.
Well, we've reviewed a number of protocols that involve pediatrics, and that generally, definitionally, is, I believe, under age 18 for just administrative purposes. So if anyone is designing a study, I think they ought to take that into consideration. So I wouldn't lump pediatrics with adult generally, because we're going to have to look at that differently than if it's just adults, obviously. And when it comes to a pediatric study, I mean, I look at those clearly, and it's basically a similar review process. I don't know that it's substantively different, with the exception that I might have to involve, since I'm an adult neurosurgeon, either one of my colleagues in pediatrics for any concerns that may be more relevant to that population, or sometimes if it's a combination situation, clearly then getting either CEDAR or CBERS involved for some of these other issues. We're seeing that particularly from CBERS for some of these replacement entities. So it's a collaborative process internally that I'll get others involved if it is pediatrics. I understand and often am asked to look at, if you will, the neurosurgical kind of issues that may relate to pediatrics versus adults. But when it comes to some of these, focused ultrasound being relatively non-invasive, it really gets into the underlying disease and whether it's a tumor or whether it's, well, usually it's for some sort of drug augmentation or something. So yes, it is a different process, but a similar process, and we'll engage the pediatric folks.
Okay. And what is, the FDA's thinking on rare diseases and, any kind of accelerated approval processes or accelerated timelines around rare diseases and developing treatments for rare diseases?
Well, at the device level, Greg, you can help me out here. I'm trying to remember the designation, but we have a couple of protocols already. I think they're called humanitarian use designation, or where they can be individual applications. So again, if it's a novel device or novel application, and we've had this like devices from outside the United States coming in, you can actually submit even an individual example protocol, for instance, or a protocol that's, say, for a population, but will be individualized, not a study of 20, 50 patients, but each time a patient presents will be enrolled in that protocol. And I believe we put that under and Greg can help me, either HUD or HDE. Do you recall, Greg?
Yeah, so the humanitarian device exemption, and Subha's just put out the link to guidance information on that. I would say we saw today an announcement that went out, FDA seeking comments on initiatives on the device side that may help accelerate the regulatory process for devices involved to treat individuals with rare diseases. So that's something just went out in the news the other day, and it's open for companies, individuals to make comment on. So keywords: humanitarian device exemption, expanded access, and breakthrough device program. Those three keywords are opportunities that sponsors may have, again, to seek to, in a more expeditious way, advance their proposed devices to the market.
Great, yeah, I just saw, and maybe it's the same thing, but there was a couple days ago a release saying that just the breakthrough status designation, there's going to be some changes around devices that address healthcare disparities and health inequities, and then also around non-addictive or products that would essentially treat pain or addiction in non-addictive ways. So essentially that, to me, implies devices that are, non-pharmacological, like potentially focused ultrasound to treat pain and addiction. I know that we'll also, from the foundation, we were going to put that in our next newsletter and comment on that, because I think that there's some interesting proposed changes there in the breakthrough pathway that could be beneficial to the focused ultrasound community. So thanks to FDA for thinking of that. Again, I want to ask folks in the audience to come up with any questions. We still have plenty of time left. I've used up the questions that I've received, but again, we have time if anyone has any more other questions. Oh, here comes someone.
Hi, I have a question about some low-intensity applications. If I have a device A and my colleague has device B, but we're both delivering the same amounts of energy to the brain, and we're interested in significant risk determination, that's device independent, is that correct? You guys care about not the device, but you care about the energy?
Did you hear that question? Yeah, okay.
Yeah, I'll take a start at that. I think that's the energy is part of it, but there's a lot more than that. It would depend on the targeting accuracy, for example, of one device compared to the other, the total exposure duration, whether you're monitoring for cavitation. So there are a lot of more subtle things. The patient population, what was the intended patient for the different applications. So energy definitely is one of the considerations, but there are multiple others as well.
Okay, with all things being equal then, but it was a device A from one manufacturer and device B from another manufacturer, those would be predicates if all else was the same? Or do you look at some of the specifics of the device?
Are you speaking that there's a device marketed already? You have to understand,
it may or may not be.
We deal with marketing. So if you have a device if there's a device on the market that we've already cleared or approved for use, and you're coming in with a second device that ostensibly is the same thing, then that's a process, regulatory process, depending on the device type, that might go 510K or otherwise. But if this is a novel device that's not been marketed, then the question is, does it have to go through an investigative protocol? And again, that's the IDE regulations, and you could look at it, but if it meets the definition or the character of a significant risk study, then it has to go through the IDE process. It doesn't necessarily meet that. We would determine that, and that's where the study risk determination comes in. But there's different contexts, whether you're talking about an already marketed device that you're comparing yourself to, or it's an entirely novel device. And as Matt said, that might factor in energy, it might factor in any number of things. From the clinical standpoint, it factors in the context of use, undoubtedly. So that's why you want to submit your study risk determination.
Thank you.
And to add to this, if you have a predicate that has been cleared for a given indication for use and given technical specifications, and you are coming in with another device for the same or comparable or identical indication for use in identical population with very comparable technical characteristics, then there is a possibility that you may argue for a technical SC. But you may want to go over that 510K flowchart and try to argue why your device is comparable to this predicate that has been already cleared, like Bennett was referring to.
Great, thanks. Dr. Frank has a question.
So I'm going to get back to Jessica's question about microbubbles, because I don't think we've really dealt with that very well. I mean, microbubble limitations right now in using with coupling with pulse focus ultrasound to open the blood-brain barrier tends to be limited in most of the trials by what the dose you would do for cardiovascular imaging, for instance. Where do you see, and so we're essentially using the bubble, a nanoparticle, or a very flexible nanoparticle for opening the blood-brain barrier, coupling with the focused ultrasound. Dosing may be very important. We can actually drop the pressures a lot, and we can actually open a lot more of the blood-brain barrier if we give more of a dose, but we're sort of running up into this limitation in some cases of this is the maximum dose based upon cardiovascular use. Can we sort of see in the future that there may be some redefinition of how much we can actually give of a microbubble, a clinically approved microbubble? I'm talking about an FDA approved microbubble for use in opening the blood-brain barrier, or actually not so much even the blood-brain barrier, coupling it with, say, sonodynamic therapy or something else where we actually deliver more drug to a tumor.
Yeah, thanks, Joe. I think we don't have the answers to those questions yet, and you know, I applaud you for doing some of the research that will help answer those questions. Blood-brain barrier opening as a low-intensity modality is different from neuromodulation because you do have those bubbles, and so one of the things that we need to understand is what is the what is the inflammatory response? How long does the blood-brain barrier stay open? There's a lot of questions there, and you know, that's going beyond the traditional thermal and mechanical damage criteria. So, you know, unfortunately, we don't have guidance on that right now, and we sort of take it on a case-by-case basis. But again, some of the things we do look for is again, the inflammatory response and the duration for blood-brain barrier opening.
You know, in thinking about that question and just some of these things where maybe you don't have all the evidence that you need, or it's still a case-by-case basis, are there things that as a community we can do together working with you all, particularly, you know, because you are also doing research yourself at OSL in ultrasound, therapeutic ultrasound. Are there things that we can do together as a community to help you better answer some of these questions so that we can get to the point of guidance, maybe more official guidance on some of these newer applications? Shuba, it looks like you want to jump in.
I can start that. Yeah, I mean, a great place to start is just to keep a track of parameters, right, that are being used. You know, there have been some great papers that have come out that have summarized the state of, you know, neurological applications and some of the parameters. But just updating that and maybe refining it would definitely help to lead to a guidance. But I think there has to be some kind of consensus as to what needs to be reported and kind of getting everybody on board.
So from the community side, if we were to, you know, kind of develop more consensus and have parameters and that we're keeping track of, and we're kind of showing those to you or engaging in discussion with you so that you have that information, that would be helpful.
And publishing them too.
Publishing, yeah.
You know, publishing them, not just for us, right? But
yeah.
For the field in general. But just as has been done for HITU, in a similar way in establishing a list of parameters that we would like to be reported in papers as well as in studies and to us.
Matt, did you have something to add? Or?
Matt,
yeah, I just to follow up on what Shuba said, I think there's a real need for a tool that can do the data mining to really produce a list of the different bioeffects as a function of the different parameters. And unfortunately, when you add bubbles, as Joe was saying a minute ago, it depends on the type of bubble and the dose. And so, you know, it doesn't take much to reach a, you know, 8, 9, 10 dimensional space. And so that's the kind of thing that we're dealing with. So yeah, I think some sort of tool that can start to at least catalog where we are now and you know, where we want to go would be helpful even before we start doing additional research.
Okay. We have another question in the room.
I had a question about a drug device. If one was developing a sonosensitizer that's a combination of drugs, and you were going to use that as a topical placement, say for a tumor, and you were using an already marketed device that's already approved as for HIFU. And my question is that twofold. One is this is a 3-drug, and 2 of which are already used intravenously, already marketed, have been for a long time. And the 3rd drug is also approved, not given IV, but would be viewed as a functional excipient most likely. My question has to do with, in terms of demonstration, will one have to prove that using these individually, or in terms of trialing these, or will they be acceptable? The goal is to put them in as a nanoparticle together, because there's a rationale that would work best together. I just was curious if you could just, in general, comment on a combination drug plus an already marketed device.
A combination drug and what was the last point?
Well, the device is a device that's already being marketed for HIFU, but it's for thermal treatments, but it's not approved for sonodynamic therapy. I was referencing
that comes under the regulations that, again, there are different subsets to the IDE regulation where there are exemptions to it. If it's clearly, if it's an approved device, but it's not being used for the approved use, then it's probably going to be subject to the IDE, unquestionably. And now, if it's being used for the approved use, but now you're doing something on the drug side that may be something different or novel, then that would probably involve CEDAR more than us. But if you look at the IDE regulations, there are two aspects to it. There may be more than two. There may be three. There's physiological research. There's non-significant risk studies, and then there's exemptions to the regulations, and there are different categories. So I personally looked at all those different categories, and they generally go through the study risk determination submission, and among the determinations could be an exemption, as maybe you're suggesting, or it's a physiological research, or it's a non-significant design. But that's the whole purpose of that submission process. Does that help you any?
Yeah, it does. I just am really trying to compartmentalize these two, and that answers my question. Thank you.
And I also just wanted to let everyone know that the various documents, the different guidance documents that have been discussed here, that have been shared in the chat, those are going, you'll be able to find those on the platform in the panel description for this session. So if you want to go back and reference any of those, they'll be there. So that's great. Any other questions? I think I'll give all of our FDA colleagues also an opportunity, if there's anything else you want to bring to the community's attention, or anything else you want to encourage engagement on with your offices, feel free to do so.
Hey, Jessica, this is Matt. We didn't talk about mathematical models, which are being used more and more these days in submissions. And I think that's just another motivation for going through the pre-sub process, so that you know, at least in our mind, whether you're using a useful model. And Dev has done a lot of work on what models are appropriate for what applications. And so that's something that we can help with, in the early stages.
That's great.
And let me just say, as the clinician, that I get presented these things a lot, and we don't profess to know hardly much, if anything. And I just did, for the gentleman that just asked that question, I just did what I often do. You can Google search your question, and it's very useful what comes up generally, because if somebody submits to me, like the previous question, that maybe they think they're exempt, I just Googled IDE exemption. And what you get is a very, very good discussion from the FDA website about IDEs, and it's called Frequently Asked Questions about Investigational Device Exemption. So I encourage not only the pre-submission process, but just to try to use the public resources that FDA has by just doing what you do, when you want to find your local Rite Aid, go to Google, and you'll find a lot of great information there, and you'll find a lot of websites of other institutions and other IRBs and everything that will be discussing that topic. And it's a learning process for me, and I think you'll find it very facilitatory if you're trying to navigate the the FDA regulatory process.
Anyone else have anything? I think also just if you have any suggestions specifically for the foundation and how we can better facilitate, either engagement of our community with you all or vice versa, or dissemination of things that you all are doing to the community, whether you have ideas now or think of them later, please feel free to always contact any of us at the foundation, and that's one thing we want to do, is to try to facilitate early engagement and more regular engagement between the community and FDA.
Jessica, I think a question has come up on the platform as well.
Yeah, that's very similar to one that we've been asked. We've kind of gone through about just combination products. So I think we've kind of addressed that. And I did
add
go
one
ahead.
more reference. I did add one more reference to the Early Feasibility Studies program at CDRH. And so we've added here a link to that guidance document that describes a lot of opportunities, again, to engage with CDRH as either, it could be new indications or as new device technology is being explored. And it really focuses on how to seek to leverage existing information and identify and add appropriate risk mitigations to allow continued development of technology as opposed to say everything all has to be done and then it's finally submitted. So I encourage investigators to take a look at this early feasibility approach.
Great. And I just want to thank all of our colleagues here from the FDA, and also thank you all for, being very open with our community, coming to forums, participating in forums like this, participating in workshops that the foundation holds, and, I think being really active in the community and engaging with the community. I know that everyone appreciates that. So thank you all, and
can I just ask you, does your website or does your, I don't know, pamphlets or whatever, do you have any sort of identification as to, just to inform, interested parties about CDRH or about our office or about OSIL? Obviously the individuals change, but the general direction of where these things will go is pretty static and stable. So I'm just wondering if you highlight somewhere, that it's, who or what CDRH is, who or what OHT5 is, or our team particularly, or Matt's team in OSIL. I mean, is that something that you're producing for your audience, so to speak?
Not specifically about CDRH, or not specifically about OSIL and the personnel. We try to highlight when there are, new guidances that come out or things like that, but we obviously don't capture everything, and we can definitely we can easily create a home on our website that has more of that information stored and readily available to our community, to make sure that they have access to that. That's a great idea. We can definitely do that. And that's
I just encourage you to do that, since I've been doing this now for 8 years, and I had a colleague who was doing it even longer. And our familiarity with your group and particularly the companies that make the devices for your sort of issues, we've developed relationships with, and I think to say to a new individual or a new institution, you have to get in touch with the FDA. That's a big place to get in touch with and have to navigate. Whereas if you identify, OHT5, I think it's a much easier starting point to get through the process, perhaps. So I encourage you to consider that.
That's great, and I
think that's a great suggestion. And Jessica, Jenna and I can help you with that. So
okay.
we can just kind of, yeah.
I think it would
show
be
you where to find this information.
Yeah, I think we can follow up and really try to help present that information better to our community so that they know where to start when and FDA doesn't look like a big black box to them. So yeah, we can definitely talk some more about that. That's a great suggestion. So I appreciate that. Okay, well I think this was wonderful. Again, thank you to all of our FDA colleagues for spending this time with us, and thank you for all of you in the room and online for joining us. And I think now we're going to have a switcheroo of some of the things up here so that we're going to have another fireside chat. So thanks again.
Thank you.
Thank you, Jessica. Thanks for the foundation. Thank you, guys.